Dear colleagues, I'm Huang X, a PhD student at Department of Critical Care Medicine, Renji Hospital, Shanghai Jiaotong University School of Medicine. I was under the supervision of Professor Worsen and Qijun Wang, the corresponding authors of our published paper in Gut. Today, I'd like to introduce our work about Enterococcus faecalis hijacks FABP2 to activate quorum-sensing signals and aggravate Crohn's disease by inducing gut dysbiosis. The gut microbiota plays a critical role in regulating intestinal barrier function, engaging in complex interactions with intestinal epithelial cells. Dysbiosis of commensal bacteria is closely associated with inflammatory bowel diseases and increased susceptibility to pathogens. However, the regulatory mechanisms between the gut microbiota and the host remain largely unclear. Establishing the causal relationship between gut microbes and intestinal inflammation is one of the major questions we aim to address. To investigate these host-microbe interactions, we developed a novel pull-down assay based on live bacteria to identify microbial interactions with epithelial cells. We selected representative gut microorganisms, including Lactococcus, Salmonella, and Citrobacter, and co-cultured them with mouse intestinal homogenates to mimic host microbe interactions. Our results revealed widespread interactions between microbial proteins and host proteins along the intestinal tract. Interestingly, only a limited set of host proteins mediated specific interactions across multiple bacterial species. From this screening, we identified FABP2 as the most important host protein interacting with gut microbes. The fatty acid binding protein FABP family consists of highly conserved cytosolic proteins involved in intracellular lipid transport, storage, and metabolism. FABP2 is highly expressed in intestinal epithelial cells, where it promotes lipid absorption and binds excess fatty acids to maintain lipid homeostasis. Following intestinal epithelial injury, FABP2 is released into the lumen and enters the bloodstream, leading to elevated plasma FABP2 levels. Therefore, FABP2 is widely regarded as a biomarker of intestinal damage. So we examined FABP2 expression in clinical samples from patients with Crohn's disease. Using immunofluorescence and Western blotting, we found that non-inflamed regions of the small intestine exhibited higher FABP2 levels than inflamed regions. We also detected elevated FABP2 levels in both fecal samples and plasma of Crohn's disease patients, indicating that chronic epithelial injury leads to FABP2 release into the peripheral circulation and intestinal lumen. Using FABP2 knockout mice in the DSS-induced colitis model, we observed significantly alleviated inflammatory symptoms in the knockout group, demonstrating that FABP2 plays an important role in maintaining intestinal barrier integrity. We further monitored fecal FABP2 levels during DSS-induced colitis and found a dynamic pattern, an early increase, a mid-stage decrease, followed by a late-stage rise to the highest levels. This trend suggests that FABP2 interacts actively with the gut microbiota throughout the course of intestinal inflammation. To clarify the role of FABP2 in Crohn's disease pathogenesis, we administered FABP2 protein orally during DSS-induced colitis, and we found that FABP2 treatment significantly worsened intestinal inflammation. Then the 16SR DNA sequencing revealed FABP2-mediated dysbiosis, particularly an increased abundance of Enterococcus species. Our in vitro experiments further confirmed that FABP2 directly stimulates the proliferation of endogenous gut microbes in a dose-dependent manner. In DSS-treated mice, FABP2 administration induced early dysbiosis dominated by Enterococcus. Using in vivo BRDE labeling, we demonstrated that FABP2 released into the intestinal lumen actively promotes bacterial proliferation. Altogether, these findings indicate that FABP2 disrupts microbial homeostasis, and gut bacteria can exploit FABP2 to enhance their own growth. And we found Enterococcus faecalis shows the strongest response to FABP2. The fish assays confirmed that Enterococcus faecalis levels were significantly reduced in FABP2 knockout mice, suggesting that FABP2 is essential for its growth. Thus, FABP2-mediated Enterococcus overgrowth may serve as a key driver of dysbiosis. To explore the underlying mechanisms, we performed the co-immunoprecipitation and mass spectrometry using FABP2 and Enterococcus faecalis lysates. We identified EF3041, a pheromone-binding protein, as a specific interacting partner of FABP2. So we constructed an EF3041 deficient strain, and we found no differences in the fatty acid profiles between wild-type and mutant bacteria, suggesting that FABP2's effects are independent of its classical lipid-binding function. The RNA sequencing analysis further revealed that FABP2 stimulation upregulated several ABC transporter genes in Enterococcus. However, these genes were significantly downregulated in the EF3041 knockout strain. This transporter system includes a key component of the OPP-ABCDF oligopeptide transport systems and the sex secretion complex, which mediates protein secretion. In addition, the GEL-E gene, critical for biofilm formation, microbial interactions, and signaling, was also affected. Together, these results indicate that FABP2 acts as a host-derived pheromone-like molecule that activates quorum-sensing pathways in Enterococcus faecalis, thereby promoting its proliferation. Given that EF3041 is required to activate the OPP-ABCDF system, we hypothesized that EF3041 deletion would diminish bacterial responsiveness to FABP2 and subsequently alleviate intestinal inflammation. Indeed, our findings highlight EF3041 as a potential microbiota therapeutic target for IBD. Targeting EF3041 may restore gut microbiome homeostasis and improve host and microbe interactions. In conclusion, our study figured out that FABP2 protein functions as a host-derived pheromone-like signaling molecule. Specifically, FABP2 promotes the proliferation of the gut commensal Enterococcus by activating its pheromone transport system and upregulating the transcription of downstream quorum-sensing signaling pathway genes. EF3041 deletion in Enterococcus or FABP2 antibody neutralization in colitis demonstrated that manipulating the gut microbiota offers a promising therapeutic approach for treating IBD and potentially preventing its onset. Finally, I'd like to give my sincere gratitude for God to give us this precious opportunity to share our work. I also want to thank Professor Worsen and Qijun Wang for their meticulous guidance and wonderful ideas, and all the co-authors for their persistent efforts. We want to thank National Natural Science Foundation of China for the funding support as well. Thank you for listening.